Now we're going to take a look at two other simple epithelia. We're going to take a look at simple cuboidal epithelium and simple squamous epithelium, and indeed see some of where these might in fact be in continuity with one another. And really the best place to see simple cuboidal epithelium classically is considered to be in the kidney. And so the slide which we're going to look at you'll find on the NYU virtual microscope by typing the word kidney into the search box and then selecting the 7th slide, or slide number 7. Now, as with the previous slide, this is a multi-tissue slide, and here it is here at lowest magnification. There are 4 separate tissues on the slide. The tissue we'll be interested in looking at is this one here, which is kidney. The reason that the kidney is such a good slide to see epithelium lining tubes on is that the kidney is actually made up essentially of millions of tubes, highly coiled and closely aligned with one another. And indeed, understanding the arrangement of the tubes in the kidney can be quite difficult until you have a little bit of experience with histology. Fortunately, we don't have to look at the whole kidney, just at one particular part of it. We're going to avoid looking in this region out here, where although there is simple cuboidal epithelium, it's a little more difficult to interpret. But perhaps after you've looked at the simpler, easier material which we're going to look at just now, you might want to return out here and see if you can see tubes lined with simple cuboidal epithelium. For now, we're going to look, to begin with, at this little piece of tissue that projects inward here. Now bear in mind that this piece of tissue is in fact a series of tubes. These ones, rather than being coiled, are quite straight. They're all compressed together, and these tubes have been cut in cross-section. So it's as if we had taken a bundle of reeds and we had chopped across the bundle of reeds, and we're staring now end on at the bundle of reeds or bundle of bamboo. And now we're going to go and take a look at how these particular tubes appear. Here we go. So here, these are our reeds or hollow tubes that have been cut across, and we're looking at them in cross-section. Let's go into even higher magnification, and here is what it is that we're going to look at. The first thing that you'll note is that there's some intense red staining, and here the basement membrane has been stained because of the stain that's used to stain the tissue. So the intense red staining lines which you see are where the basement membrane is. You'll see a lot of spaces or gaps, and these are the lumens of the various tubes which are present here. The tubes which are present are tubes which are either carrying urine or indeed some blood vessels, although we won't pay too much attention necessarily to the blood vessels. So let's zoom in to the highest magnification. And here now, if we take this tube as an example, if we were to classify the epithelium here, would we call it squamous or would we call it cuboidal? I don't know. I suppose there's a judgment call involved in this. One way that I look at it is to take a look at the overall size of the cell, judging particularly by the size of the nucleus if you can't see the cell borders. So here's one cell, 2, 3, 4. There's a 5th one here. The nucleus is out of the plane of section. 5, 6, and then maybe a 7th one, and the nucleus is out of the plane of section. And in the cells which I see here, where I can see cell borders and where I can compare the size of the cell to the size of its nucleus, we find that the height of the cell is very closely equal to what the width of the cell is. It's maybe a little bit wider. So I would call this a low cuboidal epithelium tending toward squamous. As we look here, here's an even lower epithelium. And then as we look here we've got basement membrane, we've got lumen, and in this particular one we can see a nucleus, so it bulges into the lumen, but we can't really see any cytoplasm that belongs to this cell or any other cell that might form part of the wall of this tube. So I would be inclined to call this one simple squamous. If we look over here, here's a good example. Here's a nucleus, and we can see some cytoplasm from this cell stretching out and stretching over in this direction as well. This cell is obviously longer than it is tall, so again I would call this squamous. So you could look around, you'll find epithelium which is definitively cuboidal, you'll find epithelium which is definitively squamous, and what you need to understand is that in fact many of these tubes, despite the being lined with either cuboidal or squamous epithelium, are in fact one continuous tube, and it's just that the height of the epithelium varies over the length of the tube. When we move into this region in here, or indeed into this region in here, one of the things which we'll now encounter are some of these tubes are no longer cut in cross-section, but rather they're cut in longitudinal section or in oblique section, meaning somewhere between cross-section and longitudinal section. So what I'd like you to do is to take a look in this region and see if you can find regions where you have cells that are approximately cuboidal in their appearance versus places where you have cells that are approximately squamous in their appearance. And again, you can look at the various dispositions. As we move over a little further, perhaps into this region in here, or indeed in here. Again, you look here, the cells are cuboidal. Now, in these particular set of tubes, we don't see the boundaries or the margins between individual cells terribly well. So we judge cell height and cell shape by looking at the position of the nuclei of adjacent cells. Here, in this tube, we do in fact see the borders or the margins of these cells, which makes judgment of cell shape considerably more easy. And here we see longitudinally sectioned tubes that contain or that are lined by simple squamous epithelium,